Of data that I would hand over to Mike for the analysis that he will talk about now.
Okay, so my name is Michael Gore, and I'm an associate professor in the plant breeding and genetics department at Cornell University. And I thank everyone for joining today, and I thank Sean Yarns for the introduction and John McQueen for helping out with technology. So what I'm going to be talking about in this section is about processing all of this phenotypic data that's now been collected by the high-throughput phenotyping tractor platform, and then how to properly analyze it. So here we have in an Excel format layout a geoprocessed raw data. So this is coming out of Kelly's geoprocessing computer package that he created. And so now there was some post reformatting, so it would be compatible with SAS or like R software packages. Ooh, whoops. There we go. So, one of the most important things about phenotypic data, or just quantitative data in particular, is you need to know your data. And the best way to do that is with a histogram, a box plot, or a scatter plot, QQ plot. So the quantile plot is a graphical method for comparing two probability distributions to assess goodness of fit, and generating a bunch of summary stats such as mean, standard deviation, variance with their confidence intervals, looking at the normality of your phenotypic data, testing for outliers. You can also do quantiles, and you can generate and look at their related confidence intervals, and you can do frequency counts for observations. So as like what Kelly was showing on a row by row or a plot by plot, then you can actually, quantify how many readings are there per plot. So at the speed for which the tractor was traveling, on average, there were maybe 8 to 10 readings, or like points per plot, and each plot can consist of anywhere, depending on the actual stand, from like 50 to maybe 80 cotton plants. And you can also look at or create plots to see how much phenotypic values are not present. So say if you had like a failure in one of the rows, you could then look to see if maybe some of the plots have like no phenotypic information at all, or maybe have like tenfold less phenotypic values. So that's important information to know. So I really like this quote from Parnett and Lewis, and not all outliers are illegitimate contaminants, and not all illegitimate scores show up as outliers. So the main point I'd like to convey from this is outliers are unusual data points that substantially deviate from the mean and strongly influence parameter estimates. You should always check for outliers in your data sets. If you're getting any phenotypic data sets from a collaborator or some other lab, this is one of the first things that you should do. You should always check for phenotypic outliers and then generate like the summary stats so you have an idea of the quality of your phenotypic data. And if there are outliers detected, do not ignore them. And the reason why is because this can result in several negative consequences, such as an increase in the error variance, reducing power of your actual hypothesis testing, distorting estimates of your parameters, decreasing normality if non-randomly distributed. And one important point I'm stressing here is that inferences made from these fundamentals may not be representative of the population under study. So in a sense, this is saying your conclusions or inferences that you're making from your results may not accurately reflect the population. So what are some of the potential causes of phenotypic outliers? Human errors, so this could be in phenotypic data collection or the computational code that was written to process all of the phenotypes. So this could be recording, say, with the tractor or with a Palm Pilot, or inputting manually into like a computer like spreadsheets such as Excel. So one thing that's really powerful with this tractor approach is we're not having to do any like manual imputation at all because everything is collected on the tractor and then it's geoprocessed. There can be technical errors from faulty or non-calibrated equipment used for phenotyping, so it's imperative at the beginning of every season and even partway through the season to actually investigate the quality of your equipment just to make sure that it is not faulty and to make sure it is calibrated. And so there could also be sampling errors because of different subpopulations, incorrect assumptions about temporal trends. And one thing to also keep in mind is some of these biological outliers could be actually real because there is a 1% chance of getting an outlier that is 3 standard deviations from the mean. And also there could be large gaps in the vegetation. And this is one of the major causes of phenotypic outliers in our phenotyping data sets, because when there's poor stand count, there's large gaps in the vegetation, then the soil is exposed. So in a sense, that soil is contributing noise and contamination to the phenotyping when it's actually being carried out in the field. So it's imperative to remove that noise, or your as I was saying earlier, inferences and your hypothesis testing may not be valid. So how do we detect these phenotypic outliers? So one approach that I've used in the high throughput phenotyping data sets, and others that have also used in large phenotypic data sets from maize and many other plant species, is fitting an initial linear model of fixed effects for each trait. So, possible fixed effects to include in the model are such as cultivar or line, or it could also be called a genotype. And then if there's a treatment effect such as like drought or, wet or dry treatments, in environment terms, so you may be doing like 2 or 3 locations of testing of your same genotypes of germplasm, and you may have reps, your like calendar year and the actual time when you're actually going out to the field and doing the phenotyping. So because if you're doing a trait such as like canopy temperature, it'll change over time. So this is a covariant you may want to also put in your model to look for outliers. And sometimes and often you can test for the interactions of these terms as well when you're searching for outliers. So some of the possible approaches to test for outliers I have listed here. Now there are one thing I just really want to be clear here. There are several possible approaches, and I'm not advocating any one. So if you have questions, you should maybe consult with people who are experts in this area. But I will say that these 3 are ones that work very well for my research. And in a sense, what you're doing here is you're testing for the influence and the leverage of a particular phenotypic value. And then if it's great at a certain like cutoff threshold, then it needs to be removed. And one important aspect of the detection of the phenotypic outliers is you need to repeat model fitting several times, because as you remove outliers that had maybe great leverage or great influence, there may have been other outliers that were masked, if you will. So one important aspect that folks often don't consider when they're analyzing phenotypes is often the traits are non-normally distributed. So when fitting linear models, 2 very important assumptions are that the error terms follow a normal distribution and there's a constant variance. So when your phenotypic data are non-normal, these 2 assumptions in particular could be violated. So you can use a generalized linear mixed model to analyze non-normal phenotypic data, or you can use the Beacock's procedure. And how this works is you find the most appropriate transformation that corrects for non-normality of the error terms and unequal variances. And I have a reference here for this transformation procedure. And on this next slide is some of the more common transformation procedures that you could use. And this procedure can either be carried out in, say, SAS or the R programming language. And how this, if you will, how this algorithm works, it is looking for the most optimal transformation for your phenotypic information. Now, just remember, after you transform something and you want to report it in a table, you will, in a sense, need to reverse transform it to put it back in its same values. So this is a QQ plot. So as we see here, this is comparing the normal theoretical quantiles and the expected quantiles. And what we see here is that there is tremendous skewing in this phenotypic trait here. But after we do the outlier removal procedure and we use a Bloch-Cox transformation procedure, we can then see that now this is following the expectations that we would want for our phenotypic data. And now this can be used with any general mixed model procedure to analyze the phenotypic data. So let's talk for a little about mixed linear models here. Here we have Y as a vector of phenotypic observations. Then we have a vector of fixed effects, and U is a vector of random effects, and E is a vector of error terms with mean vector and then variance. And that's, in a sense, capturing the residual variance. And then your X and Z's are incidents of ones and zeros, and you're relating your Y to your various vector of fixed and random effects. So that's just like a very brief general overview of the testing structure of a mixed linear model, which most of you have probably already come in contact with in your classes. So with a mixed linear model, you can either calculate blues of fixed effects. So a blue is a best linear unbiased estimator, and that's generated for fixed effects. And then there are BLUPs, which are best linear unbiased predictors, and those are only estimated for random effects. And all of this is in the framework of a mixed model, and this can be carried out in, say, such software programs as SAS, R, or AS-RIML. And so our first mixed model that we have here is an example of accounting for field effects. So what we're trying to do here is predict BLUPs for each line for a particular phenotype. So if you look at the random terms in this model, now the only fixed term in this model is the grand mean, but all of the other terms are random. So we have a term for environment. So also sometimes there could be, depending on the size of your field test, it could be broken across like 2 or 3 fields. And so we have a field effect there. We have a rep effect. Since this is an incomplete block design, we have a block effect. And something we also do is we account for like the grid pattern in the field, so accounting for row and column. So that helps take, if you will, takes into account some of the environmental variation that may happen in the field that could be caused by like soil. And we have a random error term for line, and then an environment by line interaction. So we could also then do this for, in a sense, blues for fixed effects, and then in the SAS programming language, we can have at least square means, and that's obtained for fixed effects. And in this particular model, we're very interested in the actual like F value, if you will. So we're testing the importance of line treatment and line by treatment interaction in relation to our phenotype. And rep and treatment and block and row and column, those are actually random terms in the model. So after we remove our outliers, we transformed our phenotypic data. So what I was saying earlier is generally there's like 8 or 9 readings per plot. So then what we have been carrying out then is we take a mean of those, say, 8 to 9 readings per plot, and then we analyze it with a weight mix model, and then using the number of observations per plot as, if you will, a covariate in the mixed model. So this seems to work fairly well when you have a very small population size, but when you have a very large population size, say of like thousands of genotypes, there's often issues where the mixed model will not converge, and so if that's the case, you may not be able to, in a sense, use this covariate in the mixed model. But this is a possible approach that should be tried first. And then, so with this phenotyping system, it can vary across space and time. So let's talk first about space. So we can fit a separate spatially autoregressive correlation structure in the row and column directions. So this is, in a sense, looking at the AR1 error structure for each environment. So this is like row by column. So it's the various row by column combinations being tested across the field. And then you can also model the correlation structure for repeated measurements of a trait on the same experimental plots over time with a variance covariance matrix. And how this works is because it's the same genotypes and the same plants that are being repeatedly measured across a particular time point for the whole growing season, or even across like 2 or 3 time points across the whole growing season, then those repeated measurements of a particular trait on the same experimental plots are going to have a correlated structure. So it's important to account for that in the mixed model. And after like all these terms are included, then you can actually do a likelihood ratio test to remove terms from the model that should not be in the model at an alpha of 0.05. And then the final model is used to estimate BLUPs for each line and to estimate variance components. So one way to look at the quality of your phenotypic data that's being collected is to calculate, say, the broad sense heritability on an entry mean level, or it can also be referred to as repeatability. So in a sense, you want to know what is the proportion of the phenotypic variation that is attributed to genes, i.e. quantitative trait loci in a particular genome or in a particular population. And to calculate the variance components, you can use the same mixed models that we talked about previous, but all of the terms need to be random so that you can derive a variance component. So if your heritability is high, then that can then tell you there is a large amount of the phenotypic variation that's observed in a population that is attributable to genes. But if your heritability is low, then a large amount of the variation that you're seeing for a particular phenotype is attributable possibly mostly to the environment, and that's something you do not want as a plant breeder. And then with these BLUPs, you can also estimate phenotypic and genotypic correlations with the with a mixed model that in a sense can be a multivariate model where you're including two or more traits at the same time to see what the genotypic correlations are. But generally, I just do pairwise. And then with a approach, you can calculate the standard errors of all these heritabilities and the correlations as well. And I've included a reference that has a link to all of the SAS code where all these calculations can be followed and carried out. So after you have processed your phenotypes, you've removed all your outliers, transformed them, and generated either blues or blups, that's when you can begin to really understand the genetic architecture of your phenotypes. And there's various approaches to carry that out, or if you want to carry out this work in a context of a plant breeding program. Now, I included more papers on these topics for further reading because each of these approaches I could spend an hour on for each one. So for the main ones, it would be like QTL mapping. So that can be either one trait or two or more traits that are correlated at the same time. And the same thing for GWAS. So that's just a different type of unrelated population design. We could do GS, so this is in the framework of a plant breeding program where you have a training set and you're actually trying to predict certain phenotypes from the genotypic information. And one area that I think is growing and is going to be actually very useful is being able to integrate plant growth models into these QTL approaches so that we're able to actually map QTLs in reference to the plant growth model over time for the whole growing season. So this is now shifting gears where I'm just going to give an example of the application of this work. And this is a recombinant inbred line population of cotton, so about 100 recombinant inbred lines, and it's hirsutum. And we have two treatments, wet and dry, and 2 reps. So if you follow my cursor here we have dry rep 1, wet rep 1, dry rep 2, and wet rep 2. And you can see the 100 or so plots for dry rep 1. You can see it's very like kind of like a light greenish color compared to this like royal blue color. So the greener it is, the hotter it is, and the bluer it is, the cooler it is. And so what we're able to do here, so this is all collected with the high throughput phenotyping tractor with the IRTs, so we're able to recreate a heat map of the field. And I'd like to call your attention to this one area of the field here. This area of the field actually had a higher clay content. So where I was talking about including an autoregressive model with row and column, so that's to help to account for some of the environmental induced variation that is, causing the actual phenotypes to change. And this is, if you will, a proof of concept. So here we have the red line, which is the population mean for the 100 recombinant inbred lines, and this is for the drought, the dry treatment, and this is canopy temperature. So the x-axis here is hour, and it ranges from 10 a.m. to 1 p.m. And on the y-axis is the canopy temperature in degrees Celsius. So this blue line here is the wet plots. So those are our control plots, and you can tell that the canopy temperature is much cooler on average for the population mean, and the black line here is the ambient air. So there is a treatment effect, a time effect, and a time by treatment effect. And this was tested in a mixed model framework. And these were our fixed effects. So the next question is, okay, we know that there's phenotypic variation, but is it heritable? Is it under the control of QTL, i.e. genes? And that's what we're calculating here. We're calculating the broad sense heritability for the plots that are wet. So those are control plots, which have a mean of 32 degrees Celsius, and that had a heritability of 0.52. And then the red colored plots, if you look over here, they had a mean of 37.98 and it had a heritability of 0.8. So these traits are heritable, and what that says is that if plant breeders wish to use this phenotype in a breeding program, it could be a possible tool. And now we wanted to link this with genotyping by sequencing in QTL analysis. So what this is for those phenotypes that I showed you, this is taking the ratio of dry to wet canopy temperature, and this is over a period of 2 weeks at 7 a.m., 1 p.m., 7 a.m., 10 a.m., and at 1 p.m. So up here, we have the chromosome position of a particular marker, and the blue here means that the QTL effect coming from the one parent TM1 will decrease the canopy temperature, whereas the red color QTLs coming from the parent TM1 will increase the canopy temperature. So what we draw from this is that over these 2 weeks and at these various time points, the QTLs seem to be acting in the same way. That is, they're always going to either decrease or increase the canopy temperature in the same fashion, but they don't have their strongest expression until 1 p.m. for each week. So that shows us that the QTLs are like turned on the strongest at 1 p.m. each week, and that happens to correspond with like the peak heat stress and drought stress for this population. So that just puts into practice kind of what Kelly showed with the geoprocessing and what I showed with trying to remove all the phenotypic outliers and transform it, and then to proceed with QTL mapping. And now Kelly is going to talk about inverse modeling. So this is taking it even further from QTL mapping.